रजिस्टर बाहर निकाल दे, इधर आ जा। दही होता है मेरा। अच्छा, जूस देगा?
ये मैं चेक करती थी, ऐसा दिखता है जी अपने कॉन्फिडेंस।
अच्छा, बहुत अच्छा करती है।
कि पता नहीं कुछ दिख भी
वो
रहा है।
हमने रोटरी न्यूज का भेजना ना, मेरी लैंग्वेज दिखा दे। एक पेंसिल दे दे बेटा। कागज और होगा, टेबल पे पड़ा होगा। सो हाँ, good afternoon to everyone. So again, we are doing good afternoon. So today I'll be starting with your unit 9, the concept of the, I'll try to take unit 9, 10, 11, 13, so that I could reach you somewhere. So
Namaste, ma'am.
Namaskar Kanchan ji. I hope you any difficulty, whatever we have learned.
No, ma'am, not any difficulties.
Okay, let's start today with the unit 9. That is
Okay,
the
ma'am.
concept of the synthetic drugs.
Okay,
So
ma'am.
main you have studied yesterday. एक मिनट ना वो दिखाएंगे 8 ऊपर पड़ा था 8 वाला 8 जी. Earlier you have studied the drugs from the natural sources. What are the natural sources from where you can get the drugs? We have taken up the unit 8 yesterday, which has focused, which has drugs which are acting on the, इससे पहले भी था मैम, we have studied till now the drugs acting on the respiratory system and essentials of pharmaceutical 1, where you have learned what are the concepts involved in the pharmaceuticals and what are the drugs used in the respiratory system. And you people have also understood all the systems, organ systems in our body. Now there is a unit 9 which focus on the concepts of synthetic drugs. So the drugs, the purpose, the main objective, the main course of this chapter is what is the importance of this pharmaceutical chemistry, what are the drugs, types of drugs, drugs obtained from natural products, what are synthetic drugs, and what is the structure activity relationship, what is the importance of this structure relation activity. So these are this chapter envisages all these concepts. So what is the importance of, somebody ask you what is the importance of pharmaceutical chemistry, which you people are studying. The very basic objective of this medicinal chemistry is to design and discover the new compounds which can be used as drugs. And even you have to design, you have to synthesize, you have to develop the new testing methods, procedures, so that any new substance you think about which can be operated on your body comes under the pharmaceutical chemistry. What are the drugs? The drugs are the chemical substances which are used to prevent or cure the diseases in humans, animals, plants, and the activity of the drug in its pharmacological effect on the subject, how it interferes with the biological processes. So, any drug, whichever we take, is a chemical substance. So sometimes it is written how it acts, it interferes with your biological processes. So, what you infer from it? That no drug is completely safe because it will act only when it interferes with your biological processes. So, why we need new drugs? When you were many years back, you have a penicillin, streptomycin, but you see now the new drugs, cipro family, the ciprofloxacins, you must have seen the new drugs are coming. So why we used to take that? Why we need it? So new drugs are constantly required as the strains, means types, the species, the different strains of microorganisms are developing resistance to the existing ones. So they are required for the improvement in the treatment of the existing disease and for the production of safer drugs by experiencing the results. So either you want to reduce or you want to remove the adverse effects. So you need new drugs constantly. What are the drugs type? Now the question comes, what are the drugs type? So drugs are mainly, there are two types of drugs. One are the drugs which are obtained from the natural source products, another are the synthetic drugs. So, what is the drugs obtained from the natural products? You know, from the time immemorial, the world is using a wide range of natural products for medicinal purpose. The products are obtained from the animals, vegetables, mineral sources, and they were very effective. It is from the 6th century BC. So, and similarly, you must have been knowing there are the Chinese emperors who had described the use of the herbal medicines. Same is in the Egypt. There are a number of medicine plants which are being used. So, in present time, even over 25% of our common medicines contain at least some compounds obtained from plants. For example, more than 5,000 alkaloids are being identified. They are present in the plants, different families of the plants, in numerous families of the plants. Morphines, codeines. Uh, so these are quinine, atropine, and there are many others like belladonna. Uh, there are, like I should say, hyoscine from belladonna. So these are the nicotine from tobacco, caffeine from coffee and tea. So there are many alkaloids which are still used and they are extracted from the plants, even in the present time. So plants are still important source of the lead compound and compounds and the new drugs. What are the lead compounds? Which is the major one through which you really work upon. Another compounds which you prepare as per the compound, they are called analogs. So plants are still important sources of lead compounds. On but on screening, identifying a material containing that active compound, the main problems are attached with it are its extraction, its purification, its assessment of the pharmacological activity. Earlier the sea sources were not used, but now the sea sources are also used. But with the passage of time, it has been found that when you are using these, extracting these compounds from the plants, either or from the sea sources, they leads to ecological imbalances. So now there is a need for the synthetic drugs, which can similar drugs which can be prepared. So what are the synthetic drugs? This wide synthetic drugs are the wide range of pharmaceutically active compounds from the plants. They were used as the starting point for the development of the drugs, and the drugs Drugs means they can be alkaloids. I told you, or you can synthesize the similar compounds. Uh, just people you are using cocaine, atropine, morphine. They are good examples of the lead compounds. So similar to these substances, new products are being formed, and which we call them as synthetic drugs. So uh, uh, so this is in screening of the substances and compounds available, are they are tested regardless of their structure. And like, for example, the screening of the soil samples lead to the discovery of the streptomycin and tetracycline antibiotics. So once you have screened out the lead compound, they have been identified. The substances of pharmacological activity or of interest, the compound which is responsible for this activity, they are isolated, and then they are used as a lead compound for the production of related analogs. So the analogs are then synthesized of most of the compounds, and they are again subjected to the screening procedure, and the sequence of the selective screening and synthesis of the analogs, they are repeated again and again, so that you can get at last the active drug. And as I just told you, the compounds on which the development is based, they are called the lead compounds, and the synthetic compounds developed from the lead are called the analogs. So the focus on the development of the new synthetic drug compounds have resulted into the incorporation of many other disciplines with the uh, like many disciplines such as biochemistry, molecular chemistry, molecular biology, medical chemistry, medicinal chemistry, these areas, and other disciplines like computer aided designing, X-ray crystallography, metabolism, pharmacokinetics, legal regulatory chemistry. So these are the and number of teams are working, which are from different fields, like biologists, toxicologists, pharmacologists, theoretical chemists, microbiologists, and micropharmacists. So the people have started for the analytic techniques to synthesize and test new drugs, so to produce the drugs which are cost effective, plus they have to be environmentally friendly. So the discovery of the new drug is a part luck and the part structured investigation. So it started with the drug and the lead compounds which are derived from the natural sources such as animals, plants, trees, microorganisms. So now I just told you marine sources which were not earlier utilized till the mid of 20th century. They are also now being incorporated. The lead compounds are extracted from them, and same analogs are prepared. So um, now natural sources are still important, but the majority of the lead compounds are synthesized in laboratory. The nature of the compounds is decided upon considering the biochemistry of the pathogenic condition. So, by which we can work on this synthetic drugs. So, in this chapter, a new concept has come into, that is the structure activity relationship. The structure activity relationship is a mean by which the effect of a drug or toxic chemical is seen on the animal, plants, or environment, and it is related to its molecular structure. This concept, structural activity relationship, it basically relates the feature of a drug or chemical structure to effect or biological activity associated with the substance. It constitutes a part of the medicinal chemistry with the modest modification in the effect or its potency of bioactive chemical compounds, which is carried out by changing their chemical structure. All the medicinal chemists, they have used the chemical techniques of synthesis to insert new chemical groups into these compounds and test the modifications and what are their biological effects. So, in this way, this is the basic assumption of all molecules is similar. Molecules have the similar, on the hypothesis that all the molecules constituting it will have the similar properties, its chemical properties and physical properties. The compound in interaction with the biological system then determines what is the biological or toxicological properties relating the chemical structure to the biological activity. So this is SAR, which is taken into account. There is a question why SAR is important. In pharmaceutical and chemical industry, yes, the process, no, importance of SAR. Uh, so, in pharmaceutical and in chemical industry, SAR means as I have just told you, the structure activity relationship. The short form is SAR. Is used to design the drugs and chemicals with commercially desirable properties. So, this has been particularly the case in the areas of drug design where chemicals with the desired pharmacological and therapeutic activities are thought of or sought. A study of the SAR of a lead compound and its analog is used to determine the parts of the structure of the lead compound which are responsible for the beneficial activity and also its side effects. So this information helps to develop new drugs and that has increased activity, a different activity from the existing drug and its side effects. So you are seeing the diagram before this, yeah, this you see the SAR is at the intersection of the biology, chemistry, and the statistics. The interlinking of this discipline have brought about the development of the research activity resembling this science of SAR. So relating structural means main aim of SAR is basically to develop an understanding of a class of molecules that are active, to find out what determines the relative activity, to know what distinguish them from the inactive classes. So SAR includes the activities ranging from the use of the expert judgment to consider the diversity of chemicals to formal mathematical associations of properties and active measures. So this involves the different fields. By this we finishes up the unit 9. Let's move to the unit 10. These are the main things you should read in that. So the 10th unit is pharmaconosis, and phytopharmaceuticals. The main focus will be the pharmaconosis, the what is the scope of this, and then classification of phytoconstituents. What are these phytoconstituents? Their extraction, isolation, characterization, and then various methods of them. So let us first understand what these words mean. Uh,
ये भी अटैच करा था साथ में, चेक नंबर भी यूटीआई नंबर भी अटैच करा था अपने साथ था।
हाँ, फिर देख लो।
या बस लिस्ट बनेगी वही।
The word pharmacognosy is derived from two Greek words. Pharmakon means drug and gignosoko means acquire knowledge of. So there is number of definitions by which the term pharmacognosy is there. Let's define that. Pharmacognosy is defined as a scientific and the systematic study of the structural, physical, chemical, and biological characters of a crude drug, along with their history, method of cultivation, and collection and preparation from the market. It can also be defined as an applied science that deals with the biological, biochemical, economic features of a natural drug and its constituent. It can also be said that it is a study that originates from the plant and animal kingdom. Because you must be understanding it is a crude drug which is just extracted. So basically, this study serves as a link between medicinal chemistry, basic sciences, Ayurvedic, allopathic system of medicine. So let's study what is the scope of this pharmacognosy. It is the applied, as we have studied, it is applied science which played a crucial role in development of different disciplines of science. The knowledge of the plant taxonomy, breeding, pathology, and plant genetics is helpful in the development of different disciplines of the science. It involves extraction, purification, and characterization of pharmaceuticals from natural resources and has signified contribution to the advancement of natural and physical sciences. Pharmacognosy is the infrastructure on which the evolution of noble medicines they are based. Crude drugs also provide essential intermediates for final synthesis of the compound. And in India, the richest source of medicinal plants are distributed in almost all parts of the country. So the cope up with the demand of the constant supply for the phytochemical industries. So, but plant drugs from pharmacognosy have answers to such questions that use of plant drugs we can treat incurable diseases. There are many drugs which are imported to India also, like balsam, benzoin. And if the cultivation is carried out in India, sufficient amount of we can save our economy. Number of diseases from which the modern science cannot cure, like stress, genetic disease, arthritic diseases, liver disorders, cancer, and AIDS. In such cases, the symptoms are treated to provide relief to the patients. So these is the study of that. So you can see what is the scope of this. The question is asked in your exam, what is pharmacognosy and what is its scope? So you can answer like that. Now, what is this? What is the classification of phytoconstituents? The constituents we get is the phytoconstituents are the therapeutic importance. They are from the plants, medicinal plants. So the medicinal potential of any crude drug, it depends it depends upon the nature of constituents present in it. The plant drugs chemically consist of mixture of organic and inorganic constituents, and these constituents are called phytoconstituents. They are formed in the plants through the activity of individual cells, and the pharmaceutically important groups, they are the organic constituents, the process by which they convert the simple chemical substances into complex organic compounds with the help of enzymes is known as biosynthesis. So things which the plants prepare, so they constitute the mixture of these organic inorganic constituents. They come under the phytoconstituents, and the phytoconstituents are of therapeutic importance, and they can be classified into the different groups like carbohydrates, glycosides, just you are seeing, tannins, phenolin, proteins, coloring matter, lipids, volatile oils, alkaloids. So let us take one by one so that I could explain you the different parts. The carbohydrates, they consist of carbon, hydrogen, oxygen, and the ratio of hydrogen to oxygen is 2 to 1. This is the wide, they have a wide occurrence in nature, and they provides means of storage and transport of energy. They are the building blocks of the cell wall. So the crude drugs, they consist of either simple or complex type of carbohydrates. The simples are the hexoses, pentoses, and methyl pentoses, and the complex, they are di and trisaccharides, and the more complex are the cellulose, starch, gums, and the basically the pharmaceutically important carbohydrates are polysaccharides if you go for the complex. What are glycosides? They are the substances which on hydrolysis means breakage with the water, they will give you one or more sugar along with non-sugar compounds, sugar compounds and non-sugar compounds. They occur in various parts of the plants like leaves, bark, fruits, and seeds. So they are further classified into different groups of pharmaceutically important. So they are also divided, but they occur in the various parts as just told. The leaf, bark, seeds also. Then comes the tannins and phenolic groups. Tannins are complex phenolic compounds. Phenolic, if you are science student, fine. If it is not, it is a benzene structure with the OH group attached. So at ortho, meta, or para, any position. They are basically phenolic compounds. So tannins are the complex phenolic compounds which are soluble in water, and they are astringent and have a bitter taste. They are classified into two groups on the basis of their ability to be hydrolyzed. First group is what are those? Hydrolyzable tannins and hydrolysable tannins. So hydrolysis, as the word say, they are readily hydrolyzed. Non-hydrolyzable, they do not hydrolyze. They are resistant to the cleavage. Cleavage means breaking. So this basically the tannins, they are used for the treatment of the burns. Because how they treat the burns? You apply it on the wound. They will precipitate the proteins of the exposed tissue which is burnt to form a protective layer, so that to stop the further effect. And They are also, you all know, in Kanpur, they are used in the leather industry. So the tanning industry for these are used for converting animal hides to leather. So these are, this is the type of some. So but as you say, there are some sugar, I told you earlier, this is of two types. So, so the second, it on hydrolysis, they give you the sugar and non-sugar part. So the example is like vanillin is a glycone of the glycosides. uh, Glycosides I have talked about of vanilla pods, and it is used, is also this phenolic groups are there, and it is used in confectionery and in the perfumery industries. You take in your homes the cloves, they also have phenolic compound. They are also simple phenolic compounds. The other are classified as proteins and amino acids. You all understand the proteins like albumin, globulin, conjugated proteins. They are the major parts of any natural products. So coloring matter, the majority like coloring, you the plants have chlorophyll which helps into the photosynthesis, some pigments like anthocyanins and the anthocyanins. So these are the coloring matters. What are we reading? We are studying various phytoconstituents. So these are the phytoconstituents. The phytoconstituents are lipids, fat oils. The lipids, they have fat oils, fats, and waxes. All come under the category of lipids. What are these fixed oils? They are liquids at normal temperature. While the fat word is used, they are solids at this temperature. So what are basically these oils and fats are? They are esters of glycerol with a long chain of fatty acids. The preparation is also like this. And these esters are also called glycerides. With these fixed oils and the fats, they are non-volatile. They are insoluble in water and they're lighter than it. They stain, if you put it on a paper, they will stain it. They are sparingly soluble in cold alcohol, but soluble in other organic solvents. What? These fixed oils and fats. They are the type of lipids. But I have also talked about the waxes. The waxes are also the lipids. And these are the esters of higher alcohols. They are insoluble in water, but are soluble in many organic solvents, and they can be sponified by alcoholic alkalis. What are these volatile oils? They are also one of the phytoconstituents. Volatile oils, आप त्रिका से बात कर लें सर, वो चेक की कॉपी दे दें सुनील त्रिका, मेरा फोन उठा लें ताकि करके भेज दें. Volatile oils are the odorous constituents which are found in the various parts of the plant. As you expose them to air at ordinary temperature, they just evaporate. And if you get some, if you like that there is a fragrance, or essence or odor of a plant, this is because of these essential oils. And these are they just evaporate, and they are present in the entire plant or any part of the plant, like coriander leaves, flowers like lavender is there, fruits, fennel is there, leaves of eucalyptus. आप लोगों ने, when you are take a steam, you people use the eucalyptus, you smell, there is essence, and this essence is due to the essential oils which are volatile. So you can just see that how it means they just. Oh, madam, check my guide. So these, you can just see these examples I am just giving you. Orange. If you smell the orange, the where are the essential oils present? In the peel. If you dalchini, what is cinnamon? It is the bark of the tree which have
हेलो
this.
गुड आफ्टरनून सर सर वो चेक भेज दीजिए जी
आप मेरे को व्हाट्सएप कर दीजिए. वी आर
सर
अटैचिंग.
ये यूटीआर नंबर रिसिप्ट भी भेजी थी अपन ने
आप बात कर दीजिए.
हेलो हाँ जी
अब
सर
चला गया ना.
अच्छा
Chemically, these volatile oils, they are the mixtures. What are these? They are mixtures of भेज रहे हैं. Monoterpenes and sesquiterpenes. They are simple hydrocarbons, alcohols, ketones. They are aldehydes, phenols. So volatile oils are therefore used as a flavoring agents or perfume in various pharmaceutical preparations, and some of them are used for the therapeutic actions as an antibacterial, antifungal, antispasmodic, and antiseptic. The plants they may serve as these type of volatile oils. They also help the plants in pollination as they attract the insects. And other constituents are.
हेलो सुनील जी भेज दीजिए
आप ना साथ एक्टिव हुआ करो मैडम. Photo constituents are the alkaloids,
सर यूटीआर
chemical
नंबर
groups
यूटीआर नंबर
present in these are the major class of chemical groups which are present in the plant drugs.
जो जब
Originally, alkaloids, why we say them alkaloids? They meant alkali like. And with course of time, the term alkaloid has altered in significance, and presently it includes many. Earlier, we used to say it is like organic basis, but now it is not only this. They includes many cyclic nitrogenous bases
जी 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 ठीक है जी
which occur in the plant. Thus, alkaloids are the naturally occurring organic substances with a cyclic nitrogenous nucleus exhibiting basic properties and a pronounced physiological action.
थैंक यू
They are also distributed in different parts of the plants. They can be in seeds, they can be in fruits. Like in belladonna, if you see the medicine, the drug, it is in the leaves. There are piper is in the fruit. Raw wolfia is in the roots, in rhizomes and the roots. There are many in the medicines which alkaloids they have. They are also distributed in different and forms the major group of chemical compounds. Resins and combinations. Resins, they are what this phytoconstituent is. They are solid or solid amorphous, means powdered structure products of the complex chemical nature. Whenever the plant secretes like exudates, and they are basically the exudates of the plants, and they are sometimes considered to be the end products of the metabolism. Resins products are produced in plants during the normal growth, or they are secreted as a result of sometimes the injury to the plants. Resins occur in a homogeneous combination with other plant products. So, similarly, there are few phytoconstituents carotenoids. The carotenoids basically you see the red, orange, yellow pigments in the plants and the animal kingdom. They are generally tetrateripoids. These are the derivatives of that, and these are divided into hydrocarbons and oxygenated forms. And they have different, like xanthophylls, they come to the category. So these are the pigments which you can see. So now in this chapter we study how we extract, how we isolate, and how we categorize. We know these are the phytopharmaceuticals. In this phytopharmaceuticals, we have to discuss the various things like extraction, isolation, and characterization. The crude drug contains the active constituent, as you have understood, which is being extracted from the plant or animal source. So these they are the active constituents which are isolated from these drugs by various methods of extraction. So whatever the drug we will form, it is because of the drugs from the so the various methods which are used for extraction and separation. So phytochemical investigation or screening basically it includes extraction, isolation, purification, and characterization, or you say identification of therapeutically or pharmacologically active plants constitution by using different physical, chemical, chromatographical, spectroscopical techniques. So let us first see the concept how we select, identify, collection, and extraction of this plant material. The extraction is commonly a technique for separation of active substance from the crude drug. We got just rough thing. Rough drug means there are number of impurities and the active substance which will be ultimately used for the drug. So there we use number of solvents. There are number of ways of extraction. So let us understand what are the extraction methods. Extraction of the crude drug can be done by various processes depending upon the physical nature of the drug and its chemical properties of the constituents which are present in it. So they were the earlier the processes which were used were infusion, decoction, digestion, but now they are absolute. Now only the maceration and precollation processes are are taken into for this extraction. So what is this maceration? It is a process which involves the immersion of the crude drug material in bulk of the solvent and for a period of time, at least 3 to 7 days, with frequent shaking. And what is percolation? In percolation, the solvent is continuously flown through the bed of the crude drug material and the extract is obtained. So, this is the two methods. Uh, so, this is then when you have extracted it, then comes the isolation and purification. Isolation and purification of constituent present in the extract, which you got it from these two methods. It also depends upon the physical, chemical characteristics of the compound to be separated. So there are some physical techniques which are employed for this purpose. These are fractional crystallization. I'll just show you how many are. These, these have come fractional crystallization, fractional distillation, fractional. You have paper chromatography, thin chromatography, and all that. So let's consider them. Fractional crystallization is useful for the separation of the volatile oil and the hydrocyanuric acid from the plant material. So this is used then. Fractional liberation. This is the one of the fractional distillation. Fractional crystallization. This phytopharmaceutical crystal out the point of they just the compound is dissolved in the and when the compound is super saturated in which it is soluble, slow concentration by slow absorption evaporation or refrigeration, the process that is involved, it crystallizes the product. Just a minute. So you have just heard the crystallization, distillation, and liberation. So the tendency, liberation, some compounds have the tendency, they are dissolved in the solvent, they just precipitate out. So this is called fractional liberalization. What is this paper chromatography? Paper chromatography, it is the principle of the component which is to be extracted. There are two liquid phases. The component is dissolved, and the extract, the component of the extract, it gets disturbed between the two liquid phases. One liquid phase is stationary, which is usually water, held in the fibers of Wattsman paper. Means there, paper means there is a paper. There's a paper, we called it as a Wattman filter paper. So there are two liquid phases. One of them is stationary, other is mobile. So the water, the stationary is the water. The water is present in the Wattsman paper because it will rise by the capillary action. And the phases, the other is mobile phase, that other solvent, other liquid, so that they will move along with that liquid. And the level which it reaches, then it is by the distance it has traveled, it and the color so obtained, we differentiate we isolate the substance. This is at the small level. So which are the, it is used for the studies of the flavonoids, alkaloids, carbohydrates, amino acids, and so on. Thin layer chromatography. So it is basically for the qualitative screening of different plant extracts. In this chromatography, the stationary phase is generally a silica gel, which is supported over a glass or aluminum plate. And mobile phase is different solvents or mixture of solvents. We just put the extract, different solvents, they move up, and the compound is soluble in one of the solvents. It will move along with this. So this is also on the small scale. Column chromatography. It is a column, or the column means a glass-like structure which contains, it is in the form of a column. It is packed, stationary phase is packed in the column, and different solvents with different polarity are used for separation of mixtures. This is basically used for the purification of vitamins, biochemicals, products, hormones, like this. Another is your high performance thin layer chromatography and high performance liquid chromatography. They also involve the same systems. Then there is that how you will now identify. You have used the methods to for isolation of the constituents. Now, how you will identify them? Identify the various chemical tests are used for the detection of the compounds like alkaloids, carbohydrates, phytosterols, fixed oils, fats, gums, mucilage. So how you detect? There are different solvents, there are different reagents with which they are tested and we identify. Like how you identify the alkaloids? The solvents of free chloroform, alcohol, water excerpts are separately stirred, and the few drops of HCl acids, and they are added and it is filtered. The filtrate, it is being filtered, and the filtrate is tested carefully with different alkoidal reagents, such as there are different reagents like Mayer's reagents, Drag-Trendroff's reagent, Hager's reagent, Wagner's reagent. So with different colors will be obtained. Like with Wagner's reagent, it will be reddish brown precipitate, and with Hager's reagent, it will be yellow precipitate. So with different, the filtrate is tested with the different alkoidal reagents. They will give the colors, and that will determine which alcohol it is. So similarly for carbohydrates and glycides, so small quantities of alcohol and aqueous extracts are dissolved separately. The filtrate is subjected to the Molich test to detect the presence of carbohydrate. Similarly, the Lieberman's, Legals, and Bontrager's test are detected to differentiate the glycosides. So these are the, like if you say detection of the fixed oils and a small quantity of petroleum, ether, and benzene extract is passed separately between the two filter papers. Oil stains on the paper indicates that it is a fixed oil. So similarly, there are many compounds now. You can read it and find out how they are being extracted, how they are being identified. I'm sorry. So there are another method which we call spectroscopy. The spectroscopy, is one of the methods which involves the wavelength of light or electrons. So let us first understand the uh, so there is a UV spectroscopy. UV means ultraviolet spectroscopy and visible spectroscopy. The it is a UV spectroscopy is an analytical technique that measures the amount of discrete wavelength of UV or visible light they are absorbed by or transmitted through a sample in comparison to a blank sample. This property is influenced by the sample composition, potentially providing information on what is the sample and what is its concentration. So, the light has a certain amount of energy which is inversely proportional to the wavelength. The shorter wavelengths of light carry more energy, and longer wavelengths carry the less energy. The specific amount of energy is needed to promote electrons in a substance to higher energy state, which we detect as absorption. So you have understood humans are able to see the spectrum of visible light from approximately 380 nm, which is violet, to as violet, and 780 nanometers, and we see as red UV light has wavelength shorter than the visible light. So therefore these concept of UV radiations is taken in the by the methods we identify. So just close कर दे क्या करती है
लेटर दे दो यहाँ बैक
absorption spectroscopy of plant constituents is measured in a very dilute solution against a black solvent by using automatic spectrophotometer, UV spectrophotometer. The colorless compound absorb in the range of 200 to 400 nanometer, it is UV range. And the colored compounds absorb in the range of 400 to 700 ml, that is the visible region. So different natural products are analyzed by this technique to which wavelength they absorb. Similarly, is the another technique of spectroscopy is infrared spectroscopy. Infrared spectroscopy is a most commonly used spectroscopic techniques, and it is employed mainly in inorganic and organic uh, determinations. The method of technique is conducted with an instrument called infrared spectrophotometer. So what is this? The infrared light interacts with the molecule. The portion of the infrared region most useful for the analysis of organic compounds, it has a wavelength range for 2500 to 16,000 nm, and the photon energy associated with this part of infrared are not large enough to excite these electrons but may induce vibration excitation of the bonded atoms which are covalently bonded. So organic compounds will absorb this infrared radiation and corresponds in the energy to these vibrations. So in infrared spectrometer, similar in process, others permit the chemist to obtain absorption spectra of compound. They are unique reflections of their molecular structures. So in this case of our plant, the infrastructure is a valuable tool, therefore, to characterize the plant constituents. The study of this reflected, absorbed, or transmit radiant energy in the region of electromagnetic spectrum wavelength ranging from zero point 0.8 to 500 nm, infrared is a functional wave number. It can be near 1200 to 4000 mm. That there are three regions of infrared. One is the near IR, mid IR, and far IR. Different wavelengths are attached to it. So by this mainly the alkaloidal components are identified with this IR spectroscopy. There are another spectroscopy called mass spectroscopy, and this mass, the underlying principle of the mass spectroscopy, it is an analytical tool useful for measuring the mass to the charge ratio of one or more molecules present in a sample. It helps you to calculate the exact molecular weight of the sample components. So by this, there is a graph which is being drawn, and the results are presented as a mass spectrum, a plot of the intensity function to the mass to charge ratio. So this deals with the electron ionization and fragmentation of molecules and the relative abundance of ions so produced. The possible structure of original molecules are then designed by the fragmentation process. बंद करना बेटा जी आपने NMR spectroscopy involves there is another form of spectroscopy, NMR. NMR spectroscopy involves the absorption of radio frequency radiations by the substance which are kept in the magnetic field. Absorption is due to the interaction of the radiations with the magnetic movement of their nuclei. Every more cell has a nuclei in the sample, and these occur at different frequencies for the nuclei with chemically different environment within a molecule. So we can just identify them by this nuclear. So these are the methods by which you just identify the different phytochemical phytoconstituents by studying the phytopharmaceuticals. By this we end up with the unit 10. Let me start with the unit 11. Unit 11 is pharmacognostical studies and herbal formulations. So, what are the main things which we are reading in this? In this pharmacoanalytical studies, it involves adulteration of a drug, reasons for adulteration, types of deliberate adulterations, what are drug evaluation, what is chemical evaluation, microscope evaluation, how you evaluate physically, and what are the different herbs which are used, what are their formulations, how you see the different herb and the drug interaction, which you must understand. This is a very important even in your own life. Adulteration of the, may I see everyone is on the line? Just I see them. I may be talking and you people may be sleeping. Hello. Yes,
Yes,
Pan-
ma'am.
yes, Ramanand, you people are
Yes.
there.
Yeah.
Under-
Yes, ma'am.
okay,
Yes, we are listening.
okay,
Yes, madam.
okay. That's
Yes, ma'am.
very nice. Very nice. It means you people are awake. It's a lunch
Yes,
Yes,
ma'am.
time.
ma'am. Yes, ma'am.
Yeah, I thought it's a lunch time. All of you
मैम
may
को
not
मैसेज भेज दो ना 5 बजे
be.
मम्मी मैम को मैसेज
So it is very important you all
मम्मी
understand. आप
मम्मी
सुन रहे हैं और आपको समझ आ रही है? Are you people understanding what I am speaking?
Yes, ma'am.
Whatever I am talking,
Yes,
you are
ma'am.
understanding? Okay.
Yes, ma'am.
Okay. Very good. Very nice. So I am taking, I have finished with the 9, 10. Now I'm moving to 11. 11 unit. I have just discussed what we will be doing. It is, first of all, there is adulteration of drug. So what is a drug basically? Drug is defined as any article which is intended for use in diagnosis, cure, mitigation, prevention, or treatment of a disease in humans, with other animals, or it is intended to alter any body function or structure of human and animals. So, what is adulteration? Then we want to study what is adulteration. Adulteration means it does not conform to the official requirements which are meant to be met by a drug. Adulteration may involve incorporation of impurities, it can be deterioration, it can be mixturing something, it can be substitution of something. Adulteration can be accidental. It does not always that somebody has done adulterated. It can be accidental or it can be intentional. So, what are the reasons for the adulteration? The reasons of adulteration, what when you feel that it be at the collection site, it can be imperfect preparation, there can be problem in the storage. So, what is in the collection? How it can be collection? Suppose it can be due to the faulty collection, as in some cases, active constituents reached at suppose in it in some season I need that active constituent and it reaches it level at a particular time of the season or a particular stage of development or at particular age. So at a collection, so at the time of collection, we may miss or we may mix something while collecting that active element. Imperfect preparation, collection of the parts may be genuine, but imperfect drying or faulty treatment may cause adulteration. During the incorrect storage, it is can be caused that there can be cause for the improper storage of the raw material of the drug. So there we can be taking into adulteration. So what are the types of adulteration? So types of the deliberate I told you there can be deliberate or it can be not intentional. So, in the type of adulteration, which are types are like confusion of the common vernacular nomenclature. You can just see types you are seeing on the screen. There can be these type of the deliberate adulterations. So let us discuss them. Confusion of the common vernacular nomenclature. Adulteration is due to the common vernacular name of different plants in different regions of India. Like for example, if we take Brahmini, it is actually to be obtained from hydrocotyl, some name of the plant. And in some parts of the India, this Brahmi is taken from another parts of the plant. And though there is a change of the vernacular means the language change is there. They consider it somewhere else and they consider it from somewhere else. So result into the adulteration. नहीं कोई जरूरत नहीं. क्रॉस सब्स्टिट्यूशन वो डाल दिया आपने
बस
Rotary
ईमेल
का.
कर मैं सारा रेडी रख रखे आप जरा चेक
अच्छा, क्रॉस सब्स्टिट्यूशन with different plant materials in place of a genuine drug, it is substituted products of the same appearance are also available in the market. You must have seen the material. We eat ashoka bark. It can be substituted with some another bark or the Indian sena. There are another barks which resemble the ashoka bark. So you can just change or substitute. It is called deliberate. जानबूझकर आपने adulteration करी. Substitution with artificially manufactured substances. So there are substances which resemble original drugs but they are artificially manufactured, like paraffin wax made yellow colored, and instead of the, and we call them as a bee wax. So bee wax, though it is a paraffin wax but added yellow color, it looks like a bee wax. So these type of things, they are artificially manufactured and substituted with the exhaustive drugs. What are exhaustive drugs? The drugs, exhausted drugs means when you take away the active substance, the remaining part we call it as exhausted material. So these exhausted materials get waste. So what the people do, they exhausted material is used entirely or its part as for the genuine, for to substitute the genuine drug. So these are the reasons for adulteration. Some addition of the foreign elements. So what naturally acting material with the drugs is excessive amounts of parts of the plant other than which constitute the drugs. So mixing the other matters. For example, limestone in asafoetida, lead shots in opium, powdered bark is frequently found to be adulterated with the brick powder. So these type of adulterations are done with the foreign matter. How you will evaluate the drugs? Drug evaluation means confirmation of its quality and its quantity, quality and determination of its quality, confirmation of its quality, determination of its quality, and its purity and deterioration of nature of adulteration. So such evaluation is carried out. Why we evaluate them? Just to understand the biochemical variation in the drug. What is the deterioration due to the treatment of storage, any substitution or alteration. So following techniques are used for the evaluation of the drugs. Morphological evaluation, microscopic evaluation, chemical, physical, biological. What is this morphological evaluation? It refers to the evaluation of a drug by its color. Morphology of anything means how it looks, how it appears. You can evaluate the drugs by color, odor, taste, size, shape, special features like by touch, by texture. So it is a technique of qualitative evaluation used on the study of morphological and your sensory profile of the whole drug. So so this is the how you feel like, that is this way you evaluate the drug. Microscopic evaluation, this is in the identification of the crude drug, it is often necessary to examine anatomical structure. It involves the microscopical examinations of the drugs. So we take the transfer longitudinal sections of the tissues which are present in the drug, and there is a considerable variation in the amount or extent of different regions of the detailed appearance of cells. So, and this becomes the criteria of drug evaluation. So this is the way it is being done. For example, the qualitative evaluation of drugs powder is done on number of features like your stomatal index, stomatal number, palisade ratio, vein islets, vein termination. Let me explain you what it means, qualitative evaluation of drugs through these parameters. Stomatal number means every plant, every leaf has a stomata. It is like a pore through which transpiration occurs, exchange of gases occurs. So, what is the average number of stomata per square millimeter of epidermal cells of the leaf? They know the number. So, by knowing the number, they can evaluate it is right or wrong. Stomatal index, number of stomata forms the total number of epidermal cells. It is the percentage. Palisade ratio is the average number of palisade cells beneath one epidermal cell of a leaf. Vein isolate, the number of veins per square meter of the leaf surface between the midway. हर पत्ते के बीच में एक सेंट्रल लाइन होती है, midway between the midrib and the margin. Vein termination number, the number of vein termination per square meter of the leaf surface midway between the midrib and the margin. So this is the microscopic evaluation. Then comes the chemical evaluation. Active constituent in a drug are determined by the chemical process. Therefore, it is called chemical evaluation. It comprises of different chemical tests and chemical assays. The isolation, purification, identification of active constituent are chemical methods of evaluation. So, this is done to evaluate the drugs. Physical evaluation. The evaluation of drug is important on the physical properties of the active constituents present. The physical standards are to be determined for herbal drugs whenever possible. They are rarely constant for crude drugs, but may they help in the evaluation with reference to the moisture content, specific gravity. These are the parameters on which it can be taken care of. Density, optical rotation, refractive index, melting point. So these are the various parameters physical evaluation at different drugs possesses. They have a different constant values for all the parameters which I have discussed, and that helps for the physical evaluation. So this is that the values so these are the various parameters which are like foreign matter, anything present in the drug which is not complying with the authentic drug is considered to be the foreign matter. Improper harvesting, deliberately addition, and improper storage, they because of this the physically the foreign matters are there. The moisture enhances the enzymatic activity. Growth of the microbes leads to the deterioration. So by this, these are the things that by which you evaluate uh, any crude, that crude means drug. Biological evaluation, the drugs cannot be assayed satisfactory by chemical or physical means. They are they are tested by the biological methods. The potency of the crude drug or its preparation, by it is being tested by its effects on the living organisms like bacteria, fungi, animal tissue, and we call it as bioassay. This method is done for the conformity of therapeutic activity of any raw material to the finished products. So this is the how these drugs adulterated and how they are isolated, extracted, and this. Now let us comes to the herbal drug formulation. This is again a herbal. This chapter, this unit also involve herbal drug formulation. There are different things in it. So WHO has now encourages, recommends, and promotes traditional herbal remedies in national health care programs. And according to the studies, 70 to 80% of people living in rural areas, they are dependent on the herbal medicines for treatment of day-to-day disease. So, you can see according to estimate, more than 700 mono and poly herbal preparations in the form of a tablets, capsules, are from more than 100 plants, and they are in the clinical use. You always knows there are different sciences of herbal science which are being used: Ayurveda, Unani, and traditional Chinese medicines. They are the important system of medicines which are largely based on the medicinal plants, and people are getting treatment through them. So herbal drugs are getting popularized in developing and developed countries owing to their natural adhesion and less side effects. So, herbal medicines are prepared from a variety of plant material, leaves, stem, roots, and bark. They usually contain active constituents which are used for the treatment. And how you classify these herbal formulations? The herbal formulations are herbal medicines or herbal cosmetics. You all know about them. You people are using them. You know about it. What are these herbal medicines? You have seen the Ramdev ji using these herbal, they are drugs out of this. So these herbs are medicinally important, and if they possesses the pharmacological activities of some therapeutic use. The herb is varied as naturopathic medicines, Unani, Siddha, homeopathic system of medicines. All of them differ in how the disease are diagnosed and which herbal remedies are prepared from variety of plant materials which are isolated from different parts of the plant like stem, root, fruits, leaves, bark. So, very common Ayurvedic formulations are, you know about it, Varthi, Anjan. These are for the eyes. Arcs, distillated. Liquids होते हैं, they are distilled essence made out of the after the drugs for soaking it and then distilling the same. Arcs, you know the asavas, they are fragmented preparations. Medicinally, alcoholic liquors are prepared by fermentation of raw vegetable juice with the honey or jaggery. They are called asavas. You have seen the churans, powder mixture prepared by mixing dry minerals, animal or vegetable substance in pestle and mortar, and that's why they just made powders, churans. And then there are gritas, that is out of the ghee. They are means the gritas, they are heated, means the ghee is heated, butter is heated on the fire, to remove water. Little turmeric juice is added to purify it, and they prepared medicines out of the ghee and oils. Vati, you all have know vati means large pills. They are prepared from pill mass. Mandha, which is prepared 12, 14 parts of water and 1 part of cereal, rice. Pia, this is just a prepared by boiling 1 pint of silver cereal in 14 parts of water. It becomes thickened and then called madam. So these are the tails. Different tails are available. Ghee and oils. They are medicated oils are available. What are herbal cosmetics? So these are the substances which enhance the beauty and increase the attractiveness of a person. They are cosmetics. Herbal cosmetics are also prepared from plants. Their products are natural drugs like animal and minerals. That the herbal cosmetics are also divided as dosage forms, organ of the body to be applied for, on which organ they are applied, dosage forms, the dosage form of cosmetics. Examples are emulsions, powder, cakes, oils, mucin, jellies, suspension, soap solution. Part of the organ of the body applied, for example, for skin, you involve powder, creams, lotion, deodorants. These are the categories of the herbal cosmetics. For hair, for nail, and you know about the nail polishes, nail removers. They are the polish removers. They are used, and like for teeth and mouth, these are the cosmetics like mouthwash, toothpaste, borderline preparations like foot powder, eye preparation, insect repellent. So these are the come under this category. So now very important topic, which is in this chapter, which is very very important. It is that how the herbal drug interaction is there. Herbal, no doubt, herbal medicine has gained momentum as people consider there is no side effect. You people have tried Ayurveda, Unani, and homeopathic medicines as the important medicine systems which are based on the medicinal plants. So they are in the they are now in the race. This herbal drugs. Roughly about 15 million people, because, but they are also taking the other drugs also. So you have to understand how they interact. According to the survey, it is estimated 18% of the patients who took prescription medicines also used herbal products. As a result, 15 million people are at a risk for the dietary supplementary drug interactions. However, the clinically significant interactions between herb and medication is unknown. But these factors, these leads the drug herbal drug interaction, mm, uh, is really a very important to know about it. What is this drug interaction? Drug interactions refers to as alteration of effect of one drug caused by the presence of second drug. So drug interaction may be beneficial or it can be detrimental. So you can just see one drug, mixing a drug you take to help you sleep and the drug you take for allergies can have slow your reaction and make driving a car or operating any machinery dangerous, because the both the drugs, they may be contradictory to each other. Suppose there is a drug food interaction. Drug food interactions result from the drugs reacting with the foods जो खाना आप खाते हैं and whatever the beverages you take. So mixing alcohol with some drugs may cause you feel tired or to slow your reaction. Drug condition interactions may occur when the existing medical condition makes certain drugs potentially harmful. So you have high blood pressure, you experience an unwanted action if you take some nasal congestant. Like if you take, you are taking for the heart as well as for the nasal congestion. So you have to see that both the drugs, they are compatible. Otherwise, it is going to harm you. So why this sort of things occur? So this is called pharmacokinetics. Interaction, this involves alteration in absorption, distribution, metabolism, or excretion of the drug. Pharmacodynamics, interaction, on the other hand, is named after the relationship between the drug concentration and the pharmacological response of a drug. So, what is this? This pharmacokinetics, how it goes about, which I was just telling you, it affects the drug absorption. It goes for the drug distribution, changes the distribution of drug resulting from the altered protein binding of high protein bond drugs have been studied intensively under this category. So these interaction really affects the drug distribution. Drug metabolism, which is called metabolism, can be inhibition or resulting into inhibition or induction. Inhibition means effect of the two substances have opposite effects on a process. For example, if we take some drug for anticoagulant effect, like warfarin, and if we take vitamin intake is increased, so it is opposite. It will decrease its anticoagulant properties. This is really very difficult. Induction, it has evolved to include any mechanism that results in increased tissue concentration of catalytically active protein involved in a drug metabolism. So these activities uh, uh, these increased enzyme activity results in a greater systematic clearance and lower bioavailability of the extensively metabolized drug. So low concentration of the drug will be available in this case as of the high metabolic activity, and you will feel there will be failure in the therapeutic use. Excretion, it is that botanically, if botanicals with diuretic effects can increase the drug excretion. So, botanical diuretics are not usually potent as and unlikely to result in significant interaction, but it will not be able to change much of the pH, but still there is an opposite effects. So there are adverse direct reactions. There can be reverse pharmacodynamics, as I was just discussing with you. These are the results of that. Because of this, it can happen. So, how we classify these drug instructions? I have just talked to you: drug, nutrient, herbal drug. So these are the three types of classification of drug interactions. So please see what is this drug interaction. As I have just told you, that drug interaction is defined as a modulation of pharmacological activities of one drug by the prior or concomitant administration of another drug. So in these reactions, the pharmacological properties of drugs can be either enhanced or diminished. Similarly, there is a drug nutrient interaction. What is that? When drugs are administered along with the food, the drug interaction may occur and can modify the activity of the drug or impair the nutritional benefit of the food. So, in most of the type of interaction, drug absorptions are effective. Food can decrease the rate of absorption of the drug or decrease the extent of absorption of the drugs. For example, vitamin K found in the green leafy vegetables, tomatoes, beef, liver, green tea, uh, can antagonize the anticoagulant activity and lowers the prothrombin time. Means it will decrease the anticoagulant property. So this is one of the examples for type of food you are taking and the type of drug you are taking. Herbal drug interactions. These type of interactions takes place when the allopathic drugs are administered with the herbal drugs. You are taking both type of drugs. Such type of drugs includes these type of interactions occur commonly in elderly critical patients, patients undergoing surgical procedures, patients with renal disease or receiving multiple medications. This happens. I am showing to give you the table. You just see the table. What you are seeing. This is a type of the herb, name of the herb, its common use, and what is its side effect or with due to the drug interaction. Epidefra, called mahang, it is used in many overcome the counter diet aids as an appetite suppressant. It is also used for asthma. So it interacts with certain antidepressant medication or certain high blood pressure medication, and there is a dangerous elevation in the blood pressure heart rate. It could cause death in some individuals. See, you have just taken a herb and you are also taken some another drug. How this certain under some antidepressant drugs. So you will see that this sort of problem may occur. Garlic, for example, garlic you all know it is many people they take for lowering their blood cholesterol. है ना? And it is also used for to decrease the triglyceride levels and blood pressure. But garlic may increase bleeding, especially in patients who are already taking certain anti-clotting medicines. So how dangerous is the garlic? You are taking in your diet. You are eating every day. Same, you can see the table I have been sharing, and it is available with your study material also. These are the herbs which people take, like ginger. These type all are the herbs which are taking. You can see how effect they are doing if we are taking the drugs as well as the other things. Similarly, in other table, you will see the interaction of the herb with the allopathic medicines. If you are taking the allopathic medicines and also taking the herbs, you will find some effects on your body or the nervous system. By this, we finish up the unit on this topic. Let's move on unit 13. Unit 13. It is on the good laboratory practices. But there are three things which are taken up in this in unit. One is the good manufacturing practices, good good laboratory practices, good manufacturing practices, and the good clinical practices. These are the three topics which are discussed in this unit. So the first of all are the good laboratory practices. So good laboratory practice is a system which has been evolved by economic or organization for economic cooperation and development. Throughout the chapter, you will see for good laboratory practice, GLP is used, and for organization for economic cooperation and development world over, it is OECD is used for achieving your goals. So these are it is a system which is evolved by this organization for achieving the goals. So this is what are this good what are this is in India, a national GLP means good laboratory practice compliance monitoring authorities established by Department of Science and Technology in 2002. India enjoys the status of the provisional member of OECD for GLP. India is observer to OECD's working group and also a member of OECD test guideline program. So India have played its place in it and has established at the national level also. They basically gives you the lines, the taglines, the timelines. They give you the system, how you have to abide by. So, what is this definition and the scope? People can ask. So, GLP controls the management for laboratories and research organizations to ensure the consistency and reliability of the results as thought about or has been outlined by OECD principles. So the this good laboratory practices, they have set of principles that provides a framework within which the laboratory functions are planned, performed, monitored, they are recorded, reported, and achieved. So they have undertaken to generate data by which the hazards and risks to the user, consumer, third party, including the world, can be assessed for the pharmaceuticals, agrochemicals, cosmetic, food and feed additives, and contaminants, novel foods, and biocides. So these are the studies have been undertaken and the guidelines are issued. So GLP helps assure regulatory authorities that the data submitted are true reflection of the results obtained during the study and therefore be relied upon when GLP. What are the powers and function of GLP authority? The main function of GLP authority is monitor the process of program implementation, establish national GLP, grants GLP certification to the test facilities based on their compliance of OECD principles, suspend, withdraw, or terminate GLP certification from its certified test facilities, constitute such other technical committees or the working groups which it deems fit to complete a particular cause for need of activity. So it constitutes such other technical committees, working groups, which they deem fit to complete a particular cause or need of activity. Ensure that National GLP Compliance Monitoring Authority operates a system in accordance with the OECD Council norms, maintains an international compatibility and mutual recognition. So these are the major aims of this GLP. What is the nature of the national GLP program? GLP certification is voluntary, which undertakes such studies either for its own purpose or for the others will be eligible to seek GLP certification. It will establish and continually update a network for GLP certified laboratories. GLP certification is valid for a period of 3 years. GLP certified laboratories shall be regularly monitored to ensure for their compliance to OECD principles of good laboratory practice and test compliance by organizing the surveillance. So this is the nature of the national GLP. NERG programs has their inbuilt feature of taking action against those test facilities which has been granted GLT and are not found to have compiled with the OECD principles of good laboratory and less guidelines which might affect the validity of studies conducted in the last facility. The test facilities interested in GLP certification would be required to give an undertaking to the national GLP program for agreeing to abide by its terms and conditions. So there are some GLP inspectors. What are this? They have national GLP program has empaneled is inspectors, the experts who currently they are employed in government test facilities organization and whose qualification experience are meeting those prescribed by the technical committees. And the function of the inspector are to evaluate the technical competence of the applicant test facility in all respects for its compliance. They are responsible for conducting inspection of test facilities based on the study and analysis of application received from the test facility. Inspectors are monitored by the National GLP office for their performance. Inspectors have access to the confidential and commercially valuable information of the test facility required for assessment. So inspectors also normally enter a test facility against the will of its management, and if there is sufficient evidence to prove that the test facility is not adhering to the GLP principles, and access to the data from the test facility is essential to protect the interest of the human health and environment, National GLP office may organize unscheduled spontaneous inspection, study audit anytime, access to the inspection team, and the records of proper inspection will be made freely available to the records to be seen. So these are the duties of the inspectors which are appointed by the National GLP. So what are the GLP principles and requirement? These are, you just see what they are. Test facility organization, test facility management should ensure the principles of GLP complied with. Its test facility ensure they have sufficient number of qualified personnel, appropriate facility, equipment, material available for timely and proper conduct of the study. Now, the second requirement is quality assurance programs. Facility should have documented quality assurance program to assure that studies performed are in compliance with the principle of GLP. Facilities, test facilities should be of suitable size, construction and location to meet the requirements of study to minimize disturbance that would interfere with the validity of study. So apparatus, material and reagents, the apparatus including validated computerized system used for generation, storage and retrieval of data should be adequate capacity. Apparatus used in a study should be periodically inspected and maintained and calibrated according to SOPs. Test systems, apparatus used for generation of physical chemical data should be suitably located and of appropriate design and adequate capacity. Similarly, another requirements are test and reference items. Records including the test item, reference item, date of receipt, expiry date, quantity received, and used in studies should be maintained. SOPs, the test facility should have written standard operating procedures approved by test facility management. They are intended to assure the quality and integrity of the data generated by the test facility. Performance of the study. The study plan should be approved by the dated signature of the study director and verified for GLP compliance by quality assurance personnel. So these are the important things. There should be reporting of the study results. Means the final report should be prepared for each study. In case of short-term studies, a standardized final report accompanied by a specific extension should be prepared. A report of principal investigator or scientist involved in the study should be signed and dated by them. And another important feature of GLP is storage and retention of records. Means all the required documents related to the quality assurance programs, personals, etc., should be retained in the archives of the period specified by the appropriate authorities. There should be proper provision. By this, we end up the GLP, that is good laboratory practices. Now the word comes as GMP, good manufacturing practices. So GMP is a term that is recognized world over for the control and management of manufacturing and quality control testing of food and pharmaceutical products. Apart from the GMP requirements, specific regulations are prescribed for various dosage forms. That is, the drugs in the form of injection, tablets, capsules, oral liquid items. So that are specific regulations are prescribed and they should be on it. GMP requires that all manufacturing and testing equipment has been qualified as suitable for the use, and their method their it also requires operational methodologies and procedures of manufacturing, cleaning, and utilized in the drug manufacturing process are have been validated to demonstrate and to be how it has to be performed. So important factors which are included with the GMP are location and its surrounding. Pharmaceutical, their important factors are what is location surrounding, what is water system, what is warehousing area, production area, ancillary areas, quality control area, personal. So there are many factors which are involved with GMP. Uh, right, so raw material, equipment, and all those materials, I'll try to switch fast through them. Location and surrounding, the pharmaceutical manufacturing unit should be situated near a clean, tidy environment free from any type of contamination, including external environment, including open sewage, drain, public laboratory. The building for manufacturing unit shall be designed, constructed, adapted, and maintained to suit the manufacturing operations so as to permit production of drugs under hygienic conditions and to avoid various uh, hazards, just as entry of insects, pests, rodents, mix up between different categories of drugs and raw material. Water system, like these are the requirements of GMP, that it shall be validated system for treatment of water drawn from own or any other source to render it portable in accordance with the standards specified by Bureau of Indian Standards of local municipality. And so water so produced shall only be used for all operations except washing and cleaning operations where portable water may be used. Water shall be stored in tanks which do not adverse effect on the quality of water, ensure freedom for microbiological growth. Tanks should be cleaned periodically and records maintained by the licence in the behalf. So, licensee in the behalf, the disposal of sewage and the effluents from the manufacturing shall be in conformity with the requirements of the Environment Pollution Board. So, this is about the water. And there are the warehousing area. You can understand well, warehousing area are designed and they are adapted to ensure good storage conditions. They shall be clean, dry, maintained with acceptable temperature limits where special storage conditions are required. So there has to be a proper housekeeping and rodent pest and vermin control procedures and records to be maintained. Production area. So working and process space shall be adequate to permit orderly and logical positioning of equipment and material and movement of personnel to avoid cross contamination and to minimize the risk of omission or wrong application and manufacturing and control measures. About the ancillary areas, rest and the refreshment rooms shall be separate from other areas. The area shall not lead directly to the manufacturing and storage areas. Facilities for changing, storing clothes, and for washing and toilet purposes should be easily accessible and adequate for the number of users. And the toilets separate for males and females. They are and should not be directly connected with the storage areas. So this is something about the ancillary area, quality control areas. The design of the laboratory shall take into account the suitability of construction material and ventilation. Separate air handling units, other requirements shall be provided for biological, microbiological, and reduced isotope testing areas. So it's very important. Personnel, prior to employment, all personnel should undergo the medical examination, eye examination, and we should be free from the tuberculosis, skin, and other communicable or contagious diseases. Thereafter, they should be medically examined periodically, at least once a year. So, all personnel shall wear the clean body coverings appropriate to their duties. And before entering into the manufacturing unit, they should change rooms separate for each sex with adequate facilities for personal cleanliness, such as wash basins with running water, clean towels, hand dryers, soaps, disinfectants. So, so these are the take care should be taken with the personnel who are employed with the industry. What is there about the raw material? The license shall be keep an inventory of all raw materials to be used at any stage of manufacture of drugs. They should maintain the records as per the schedule. All incoming materials should be or shall be quantized quarantined immediately after receipt or processing. All material shall be stored under appropriate condition and in the orderly fashion to permit batch segregation and the stock rotation. Equipment shall be located, designed, constructed, adapted, and maintained to suit the operations. वो कल वाला पड़ा है वो पापड़ी वाला कल भेज दिए खराब हो जाएगा. Maintained to suit the operations to be carried out. Each equipment should be provided with a logbook wherever necessary. Balances and other measuring equipments of appropriate range, accuracy, and precision should be available in the raw material stores, production, and the process control operations, and these shall be calibrated and checked on a schedule basis in accordance with the SOPs and records maintained. Documentation and records are also important in GMP. That is, documentation is an essential part of the quality assurance system. As such, it may be related to all aspects. Good manufacturing practices, they usually aim to define the specification of all materials, method of manufacture, and control to ensure that all personnel concerned with manufacture know the information necessary to decide whether or not to release the bath of drugs for the sale or to provide audit trail and shall permit investigation and the history of any such item. Records shall be made or completed at time of each operation in such a way that all significant activities concerning the manufacture of pharmaceutical products, they are, you can trace them. So another very important feature are the labels and the printed material. What are the use of the labels? You all see the all tablets or the so the labels are absolutely necessary for identification of the drug. Their use, the printing shall be done. The bright colors, they should be legible, and they should carry all prescribed details about the product. Quality assurance, like it is a totality of the arrangements made with the object for of ensuring that products are of quality, they are required for the intended use. The system of quality insurance appropriate the manufacture of the pharmaceutical products, and it has to ensure that the pharmaceutical products are designed and developed in a way that is in accordance to the requirements of GMP. So these are other important features are quality control system, product containers and closures. Like containers and closures, you shall comply with the pharmacopoeial requirements, suitable ventilated test methods, sample size specifications, cleaning procedures, sterilization procedures should be taken care of. Master formula records, manufacturing. You should know that the master formula records relating to all manufacturing procedures of each product, batch size to be manufactured, and they shall be prepared and they should be endorsed by head of production and quality control. The master formula includes a number of things: name of the product, patent or the proprietary name, quantity, and reference number of all materials are the statement of expected final yield, statement of processing location, the principal equipment used, detailed stepwise processing and instruction of in-process control, any specific precautions used, any packaging details and specimen labels, and so on. So these are the basically the master formula records which are being kept when they are being manufactured in a lab. So there should be a packing records which should be kept each batch wise or part batch and the batch wise, and they should have relevant records for packing instructions and the method of preparation of such record to avoid any transcription errors. Product recalls, complaints and adverse reactions which you receive, they should be properly reviewed, record according to the written procedures. Specific requirements means sterile products, various critical, sensitive in nature, a very high degree of precaution, prevention, preparations are needed. So these are the specific requirements to be taken care. Aseptic areas which are very important to make them free from microorganisms whenever it appears. As far as the garments are concerned, the garments which are used by the personnel working in the aseptic area shall be of special type. So while in GMP they have to be taken care of. So these are the GMP requirements which are being taken up in this unit. So the last part is the good clinical practices. Good clinical practices we pronounce by GCP. They are the set of guidelines for biomedical studies which encompasses how to design, conduct, termination, audit, analysis, reporting, and documentation of the studies involving the human subjects. So, so what are there are main principles of GCPR. Clinical trial should be conducted in accordance with the ethical principles. Before a trial is initiated, foreseeable risk and inconvenience should be weighed against the anticipated benefit for the individual trial subject. Right, safety, and well-being of the trial subjects are important when you take this GLP. Available non-clinical and clinical information on investigable products should be adequate. So clinical trials should be scientifically it should be sound. Trial should be it should always be conducted in compliance with the protocol that are received to the by the institutional review board. Medical care is given to, and the medical decisions made on behalf of the subject should always be the responsibility of a qualified physician. Each individual involved in conducting a trial should be qualified by education, training, and experience. Freely given information, consent should be obtained from every subject prior to the clinical test. All clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting. Confidentiality of the records could be that could identify subjects should be protected, respecting the privacy and confidentiality rules. And investigational products should be manufactured, it should be handled, stored in accordance with the good manufacturing practices, systems, and procedures of quality of every aspect of trial should be implemented. So you just see with the with this you have now there are certain you have gone through the this good clinical GCL and there GCP is an so compliance with this standard. GCP in nutshell you will understand is an international ethical and scientific quality standard for designing, conducting, recording, and reporting trials that involve the involvement of human subjects, and so we have to comply with it. And now you will be also studying that compliance with this standard provides public assurance that the rights, safety, and well-being of the trial subjects on which they are tested are protected, consistent with the principle they have their origin. What are those principles? They have their origin in Declaration of Helsinki, and that the clinical trial data are credible. So these are the concepts which you will be going through, and the important thing is secret vectors international. So this way you will be you will read the various contract research organizations sponsors schedule Y of drug and cosmetic acts which usually deal with this clinical studies. So institutional review boards, independent ethics committees are also there in this unit, which you, when you will be reading, you will be going through. So by this, we usually finish the unit 13. I feel, is any difficulty there? If there is any difficulty, you can just ask me. I have covered the major portions of this course MVE 001, which will give you a very wide image of pharmacology, your whole organ systems, the basic unit of organ system. So almost we have covered almost all the chapters. Even then, if you have some problem anytime in understanding that, you can just post on my email your problems, that is kiranangra@gmail.com. I will reply you back. If any, in case you have any issues for understanding any problems, you can ask me. So I wish you all a very good luck. Any problem, I am here to take up. I am finding the strength of students more today. So any problem, you can just talk to me and you can just. I wish you, all of you, best of luck, and may God help you to take your exam well. Right? So you should note down my
Thank
email, kiranangra@gmail.com.
Yes, ma'am.
So thank you to all of you for bearing me for 4 days, listening me, understanding MVE 001. Thank you very much.
Okay, ma'am. Thank you, ma'am.
Is there any problem? Any problem to Rahul Sharma? Any doubts? No
Nothing,
problem.
ma'am, nothing.
Problems means
No, ma'am.
in understanding some issues. Prathaji,
No, ma'am, no issues.
thank you.
Okay, thank you,
Tanya,
ma'am.
thank you. I wish you best of luck to take this paper, this course MVE 001 with the flying marks. Thank you very
Thank you,
much.
ma'am.
Thank you, ma'am. Thank you very much.
Thank you, ma'am.
Thank you very much to everyone. Now it's fine. बस
यस मैम, आज तो काफी जने थे, 2 4 5 6
चलो
मैम ये ना जी,
इसका हो गया, इसको पहले आप हम इसको
हाँ,
ये सॉरी इसको लेट मी गो फ्रॉम हियर.